we're back, and now that we've kind of talked about the different parts of the microscopic anatomy of skeletal muscle, we're going to get into a little bit about how does these muscles actually go about contracting. Compared to what we've been doing, this is probably one of the tougher physiology concepts we'll deal with this semester. So a little bit more in depth than a lot of the different types of physiology we've talked about so far. So the main thing to kind of realize is that when it comes to stimulating a skeletal muscle, we're going to be doing this with the nervous system, and any particular muscle cell or fiber, in order to be stimulated, is going to have some type of connection with an actual motor neuron, so some branch of a motor neuron. What we're going to see is we do have certain motor neurons that, as that axon travels down, it will branch out into a number of different areas, and pretty much any single motor neuron, and if it branches out, let's say it goes to 4 different other areas, all those 4 muscles that are under the control of that single motor neuron, let's say, those are what we consider a motor unit. It's going to be that motor neuron and all the muscle cells or fibers that it connects to and is therefore going to control is a motor unit. We'll talk about it a little bit later on, but you're going to see that certain muscles, the motor units can be quite large, maybe 1,000 cells per unit. In other areas where we have a little bit more dexterity, you might have one going to 4 different muscle fibers. And again, this really depends on the muscle. So small motor units, like it says, they're less than 5 fibers. A lot of times we'll see those in the hands, on the face, where you have this really fine control. Much more postural muscles, like things like the bicep or the quads, those are going to have much larger motor units, which might have several thousand cells per motor unit. Again, how many fibers are in a motor unit, it's kind of opposites on how fine that control is. So the fewer fibers there are in a particular motor unit means we have finer control over that. The other thing we'll see is when we start going to talking about neurons that are controlling this in the brain, the other thing we'll see is the smaller the motor units are, the more neurons we have to have in the brain managing the control of that. But small motor units are going to have greater control, larger motor units are going to have smaller control. And again, the other thing is a lot of times these fibers are spread throughout a muscle and not in one particular area, which means we can, when we're doing just a single motor unit or a few small motor units, it's not going to just be contracting one half of the muscle or one area. You can get a weaker contraction throughout the muscle. So these are kind of spread out. And this also helps when we have to do a sustained contraction. You can rotate through motor units and keep that muscle contracted, even though certain fibers are fatiguing, and then being able to recover while you're maintaining that contraction. Where these neurons hit a muscle cell is what we refer to as a neuromuscular junction. They did not get particularly fancy on the name of that one, but this is that little area where you're going to have the end of that axon going up in very close proximity, but not generally touching the the muscle cell. So you have the synaptic end bulb, what's called the motor end plate, the part of the muscle cell that's right across from that, and then you have that space in between, which is the synaptic cleft. And again, for this to work, what we're going to see happen is all the sarcomeres that are in a particular muscle cell are going to contract together, and this will overall shorten that fiber, and if we do enough of those, it will overall shorten that muscle. What we're going to see is this is a process that involves a number of steps. We're going to talk about what we refer to as excitation, how we stimulate the muscle, how we connect that stimulation to the contraction mechanism, that's that excitation-contraction coupling. And then we get into contraction. After we talk about all that, then we can talk about how does the muscle go back to a relaxed state. And this is a nice little overview of all the events. What we're going to spend our time on right now is getting to that step 1, what is happening to stimulate that skeletal muscle fiber. So we're going to start with that one. So the first event that happens in muscle contraction is going to have a motor neuron sending an impulse down to the end of that synaptic end bulb and causing the release of a neurotransmitter, abbreviated here as ACH. That stands for acetylcholine, and that acetylcholine, once it's released from the end of the neuron here, it's going to spread out, go across to the motor end plate, and bind to receptors there. How this is brought about is it's actually when that nerve impulse travels down, and when it gets to the end of the synaptic end bulb, it actually causes what are referred to as voltage-gated calcium channels. So these are gated proteins that when the voltage changes on the membrane, it's going to cause those to open up and let calcium flood in. This calcium flooding in, there's a whole cell biology type of thing going on there, but this calcium going in there will bind with proteins that cause these vesicles to fuse with the membrane and dump out that acetylcholine into the synaptic cleft. When this happens, you're going to get a bunch of exocytosis of that acetylcholine, a bunch of vesicles dumping this acetylcholine into the synapse. And again, we know diffusion and how that works. We dump it out of there on that one side. It's going to move from areas of high concentration to low concentration. So very quickly, it'll move across from here over to the other side of the synaptic cleft and is going to bind to receptors there. When it binds there, we're going to see this is going to cause a change in that muscle cell. And what we're going to get into now is how that happens. But you can see here's the basic idea of that excitation. You have a nerve impulse traveling down this axon right here. It causes an opening when this nerve impulse reaches these voltage gated channels, causes calcium to flood in. It causes these vesicles to merge with the membrane, dump acetylcholine into the synapse, and that acetylcholine is going to go across and bind to acetylcholine receptors on the muscle cell. So what we need to do now is we need to back up a little bit and talk a little bit about what is going on in muscle cells in general. Because for us to understand what happens with this excitation and then what happens with the excitation-contraction coupling, we need to understand a few basic facts about the resting state of muscle cells and nerve cells both, actually. What we're going to see is muscle cells are one of those that have a resting membrane potential, is what we refer to this as. So there is an electrical difference on the inside of the cell membrane or the sarcolemma and on the outside. What we're going to see is extracellular fluid, because of what is going on in these cells, is much higher in sodium ions, while the intercellular fluid is much higher in potassium ions. The plasma membrane is impermeable to sodium, so once it gets out, it's going to have a hard time getting back in. It's a little leaky to potassium, so we have some of that leaking out, and we're going to constantly be pumping it back in along the way, pumping more sodium out. What this kind of does, though, is because of these sodium-potassium pumps that we mentioned back in the cells chapter, the sodium-potassium pump will pump 3 sodiums out for every 2 potassiums it pumps in. Given enough time, what this is going to do is it's going to give you, because the membrane is completely impermeable to sodium ions, you're going to get a large amount of sodium ions out on the outside and some of these potassium ions that can leak back out. Overall, you're getting a lot more positive ions on the outside of the membrane than on the inside of the membrane. And what this is going to lead to is a polarized or charged membrane. If we were able to put little voltmeters on each side of the membrane here, we would get a voltage difference across this membrane. So the inside of the cell is going to be more negative relative to the outside of the cell. This difference across this membrane is what we refer to as membrane potential. The membrane potential is that electrical difference across there. The one thing we do know with this a little bit is that because we have all these ions of sodium on the outside and all these potassium ones on the inside that are leaking and getting pumped back in, we know if we gave them an opportunity, though, they're going to try to go with their concentration gradient. So if there's a lot of sodium on the outside of the cell and all of a sudden you make the cell permeable to it, what's going to happen to that sodium? It's going to try to flood into the cell. Same thing with the potassium going out. The difference here, if we were to actually put a voltmeter across, is about a negative 80 to negative 90 millivolts in terms of that membrane difference or that membrane potential. Again, I'm not going to give you a multiple choice that is asking for the exact millivolts on this one, but I want you to have a basic understanding of what that difference means for us. The other thing with this membrane potential, as I was already kind of saying, is if you can open up the membrane to these particular ions that it's normally impermeable to, you can get a change in that membrane potential. So by opening up a sodium gate, for example, you're going to have a big influx of sodium ions. If you opened up a potassium gate, you'd have a good big efflux of those ions. What that does if you open those up is you're going to get a change in that membrane potential. And if we can do this in response to a stimulus, what we've done to this cell is we've actually excited the cell. So again, Excitability, as we kind of mentioned in the first one here, it is this property that says the cells can respond to a stimulus. What we're going to see is there is different gated channels on these muscle membranes. We have two main types that we're going to worry about. There is mechanically gated channels as well. These respond to pressure and sometimes will open in response to those. I'm not too worried about those and having you focus on those. What we do have to worry about though is what are referred to as ligand gated channels. These are channels that when a particular molecule binds to that gate, it will open up that gate. In the case of muscles, we're going to see there is definitely ligand-gated channels, that the ligand is that acetylcholine. When acetylcholine binds to these ligand-gated channels, it will open them up, and whatever they are specific for will be allowed to flow in or out of the cell depending on its concentration gradient. The other type of gate, and we've mentioned it a little bit with neurotransmitters already, are voltage-gated channels. What these respond to is a change in that membrane potential in the surrounding membrane. So in this case here, these are a lot like a temperature on a thermometer. When it hits a certain number, it's going to cause a change. And it's like with a heating system, when the temperature gets down to, let's say, 65, that might cause, at that point, that switch to trip that says turn on the furnace. In the case here, we're going to have a voltage-gated channel that may respond to a change in that membrane potential. It's normally negative 80, negative 90. We might get something on this one where if it gets down to negative 55 millivolts, that will cause these voltage-gated channels to open. And then again, whatever they're specific for can flood into or out of the cell, again, depending on its concentration gradient. So again, if we go back to the muscle cell now, we have these neuromuscular junctions that are being supplied by this particular motor neuron at these neuromuscular junctions. And we've talked about the fact that when that info comes down here, that motor impulse, it is going to get down to the end of the synaptic end bulb and cause a release of the neurotransmitter acetylcholine. This can flood across and will actually bind to receptors on the other side. What's interesting about the acetylcholine receptors on the motor end plate, or on that area right across from the motor neuron, is that these are actually gated channels. These are ligand gated channels. The acetylcholine, when it binds to it, is going to cause a sodium channel to open up. We know that there's more sodium on the outside of the muscle cell than there is on the inside of the muscle cell. So when you open up that channel, what's going to happen is those sodium ions, because of their high concentration on the outside, are going to flood into the cell. That is going to change that membrane potential. This is the start of the excitation process. So again, flooding across here, binds to acetylcholine receptors that are over here. These are connected to these sodium channels. These ligand-gated sodium channels, when the acetylcholine binds to them, are going to open up. You're going to start getting sodium flooding into the cell. It's going to bring about a local change in the membrane. So this is only happening where that acetylcholine is binding to these particular sodium channels that are gated by that. Where we find these in the muscle cell are only at that motor end plate, right across from the motor neuron. You're going to have gated channels, ligand-gated channels that respond to acetylcholine that are going to let sodium ions in. Again, you're going to get a local change in that membrane. So it might have been at negative 80, let's say. It starts going to negative 70. It starts going to negative 60. So we're changing that membrane potential at this motor end plate. What is interesting about muscle cells is in the area surrounding, so if this is our synapse right here again, in the areas right at that neuromuscular junction, you're going to have a lot of ligand-gated channels. Directly next to that motor end plate, we are going to have voltage-gated channels for both sodium and potassium ions. These are going to respond to changes in that membrane potential. So in this case, their value that will turn them on is about negative 50 millivolts. So if we get this local depolarization happening and more sodium ions go in, more sodium ions go in, it goes from 90 to 70 to 60. If it hits 50, what that means is the voltage-gated ion channel, the voltage-gated channels right next to this for sodium, they're going to open up. That is this threshold value. If we get to this threshold value, it's going to cause those voltage-gated channels to open up. What is interesting on this muscle cell is the only place where you find the ligand-gated channels is at that motor end plate. All around the rest of the cell, so the entire sarcolemma, down into the T tubules, throughout all the rest of this membrane, you are going to have these voltage-gated channels. And like it says here, how much depolarization happens depends on how much sodium goes in at that motor end plate when we get the acetylcholine binding. So if we're talking about a muscle and whether or not we contract the muscle, the first thing we have to do is, if we get that acetylcholine being released and it causes a little bit of sodium influx, but not enough to reach the threshold value of those voltage-gated channels, what's going to happen is nothing. You're going to get a little bit of sodium flowing into that cell, but if it doesn't reach that threshold value, it's not going to open up a voltage-gated channel. If no voltage-gated channels open up, you're not going to be able to connect that what is happening at the neuromuscular junction to the actin and myosin fibers further down in the cell. If it does reach threshold, what's going to happen is that first voltage-gated channel right next to the motor end plate, it's going to open up. But what is interesting to this, if that one opens up, you're going to get ions flooding in both directions. It's going to depolarize the membrane there. There's another channel right next to that one that's voltage-gated, and another one next to that. And what happens is once you start these voltage-gated channels opening, it's kind of like having a bunch of dominoes set up, and once you tip the first one over, it knocks over the next one, which knocks over the next one, which knocks over the next one, and you get this traveling, in the case of dominoes, this traveling tipping over happening. In the case of the cell here, we're going to get one voltage-gated channel opening up, which triggers the next one, which triggers the next one, so we're going to have this electrical change spreading out and covering the entire membrane of this muscle cell, over the entire sarcolemma, down the T tubules. And that is this idea of excitation, and as it starts traveling, that's going to get into this excitation, contraction, coupling. There is a few things that can happen that is kind of interesting, some autoimmune diseases that can be an issue that mess with this. And in terms of myasthenia gravis, this is an autoimmune disease where you get antibodies that bind to the the acetylcholine receptors, and then those receptors get removed because of this antibody binding to it, which means in people like that, they get really weak or very minimal muscle contractions because they cannot cause excitation of these muscles. So a lot of times you're going to get paralyzation of eye and facial muscles with that one. So what we're going to pick up with the next time, so we've got through this idea of what we've done right now is basically got through the idea of excitation. So how, when we send that stimulus down the neuron, it will cause the acetylcholine release. That acetylcholine release can cause a change in the membrane potential at the neuromuscular junction. If that reaches threshold, that's going to move us to the next step, which is the excitation contraction coupling, which we will talk about in the next video. So for now, we'll stop right here. In the next one, we'll pick it up and move from this excitation process to how it's going to link to the rest of the contractile process.